Hey everyone, welcome back. We're going to get right into how we help in maintaining chromosome numbers during mitosis. So, a chromosome is a condensed form of nucleic acid DNA that is called chromatin. So, chromatin is the uncondensed form of a chromosome. I'm going to actually do blue in this one. Chromatin is unwound and relaxed in the nucleus, and it's in this form where replication takes place, okay? And actually transcription and translation also, but we'll talk about that later, okay? So once the cell has replicated its DNA, it's then going to condense into the structures we call chromosomes. So each species has its own set and number of chromosomes. The term diploid means a complete set of DNA consisting, just write consist because that's, I can't, if I put ing it's going to look funny. Consisting of one copy of each chromosome from mom and dad, okay? So we get a complete number of chromosomes. 1/2 comes from mom, 1/2 comes from dad. So for a human, we have a diploid number of 46 chromosomes. "23 came from mom and 23 came from dad. So haploid is 1/2 of the DNA then. A haploid number is 1/2 of the DNA. And so this is what we see in sperm and egg or in sex cells. You get half the DNA. So for a human egg or a human sperm cell, that would be 23 chromosomes. One of each type of chromosome, okay? And so when DNA replication occurs, we produce a chromosome consisting of 2 sister chromatids. And so I'm going to kind of draw that over on this part over here, just so you can see it. And then on the next slide, you'll see it even better, all right? So I'm going to use blue as one chromosome. So we'll just say this is one parent's chromosome. And there's another one from the same parent. And then I'm going to use red for the other parent's chromosome. And there's the other one, okay? So you can call either side from mom, the other side from dad. When replication occurs during that S phase in interphase, so remember, during the S phase, synthesis phase, is when DNA replication occurs. Each of these chromosomes is going to replicate. And so what we're going to get is this. We're going to get And so I'm just drawing, this is that this chromosome replicated, and then they attach. And there's where they're attached, okay? Let's go back to that blue. We'll do this chromosome. Looks like little K's, but you know, it's that one, right? And then we got this one. There's the attachment for this one, boom. And go back to that red really quick so you can see the attachment for the red right there, okay? So each of these chromosomes is now consisting of 2 sister chromatids. So this is a chromosome consisting of 1 chromatid. This is 1 chromosome consisting of 2 sister chromatids, and they're held at a region known as the centromere. So that's the centromere. And so let's look at what it looks like when you can actually draw. So here's a prettier picture. Here we have that single sister chromatid, and here we have that replicated chromosome with 2 sister chromatids. They're held in place at the centromere positioning right there, okay? And so in our cells, in human cells, we have 46 chromosomes going through this. Whereas in the slide right here, this would have been 4 chromosomes going through this, because this is the 4 chromosomes, and this is still 4 chromosomes, but they're all duplicated down here, okay? And so here you see, again, a cell with 4 chromosomes. They used red and blue as well, because those are just easy to see separately. And then each of those chromosomes in S phase, during the synthesis phase of interphase, they get replicated and they're held together. The sister chromatids are held together at the centromere. And this is what is going to allow us to control the DNA during mitosis, so we can pull the DNA apart. Each of these individual chromosomes is going to separate from its sister chromatid so that we get identical copies on each side. So I'm going to stop here and we'll get into mitosis in our next video. Bye.